Hey, welcome to another episode of the Traverse Science Podcast. Today we have a repeat guest, Dr. Kelly Swanson, professor of animal and nutritional sciences at University of Illinois at Urbana-Champaign. And we talk a lot about fiber today. What is the definition of fiber? What are the structural components of fiber, the different subtypes of fibers, functional characteristics of fiber, clinical trial design, pitfalls in publishing. One time Kelly went and confronted someone at a conference for not describing fiber well enough. So this is great. Even if you're well versed in fiber or not, it's a great place to just refresh, get the basics of fiber, and hear it straight from someone who studies this day in and day out. So enjoy. Bringing it back, maybe especially more into your domain a little bit with pet food and dogs and cats. What are like the top considerations someone should have between feeding fiber to a dog versus a cat? Right? They have extremely different diets, right?
Yeah, well, they can be similar, but when you get to fiber, the cat certainly is more sensitive, especially with fermentable fiber. And I still give presentations. I refer back to a publication, I think it was 1995, that out of the Fahey lab, when they first were kind of getting into fiber with pets, and they compared fecal inocula from cats, dogs, pigs, I think it was horses, cattle, and humans, all in a couple of papers. And if you look at the microbes from the cat, they were just as active as that from any of the other species. So they're a carnivore. But I think the big thing is their response, the host response to all those short chain fatty acids and just fermentation in general is very different. So yeah, when you think about formulating cat foods and versus dog foods, there might be some things you say, okay, they can be very similar. When it comes to fiber, the tolerance is lower, so you'd have to be careful there. They still are unique. I see you're like, oh, I can see you thinking there. The cats and dogs are still unique, so they still have different requirements. But some things you can, in pet food, the other thing too, versus livestock, when it comes to fat and protein, like amino acids, fatty acids, typically we're not close to the recommendation anyway. They're usually kind of overfed protein and fat. So that's where the composition can look quite similar, just because cat foods will still usually on average have more protein. They might have a little bit different format and more animal based ingredients than dogs that are more omnivorous. But when you get to fiber, the threshold is going to be different, so you have to be careful there with the cat. You can overdo it with dogs as well. Just too much soluble fermentable fibers can lead to loose stools, so you have to be careful there.
Why is there fiber at all, actually? Why even care about fiber in cats, right? So if they're carnivores,
Yeah.
do they have any... I'm not going to use the word need because that's a little loaded term, but
Yeah.
like would a natural
it,
cat... Oh, maybe this is your point about eating insects too. What is there fiber in a carnivorous diet?
there is, and that's where most of it will come from the animal fiber. Where if you think about, and this is where people argue too. So yeah, we've tested raw foods before in cats actually, with using the domestic cat as kind of as a model for zoo cats. So the large cats and tigers and jaguars and stuff like that. We did that work over a decade now, where we're looking at some of these raw meat diets where there was virtually no plant products at all, and they were quite constipated. The large cats actually did better than our domestic cats did. But in some of those, if you think about our domestic cat, maybe some of the bigger cats too, but if you think about domestic cats, if they're eating mice and things, usually they eat the whole, right. They're not taking out just parts of the carcass, they eat the whole carcass. Where
they're not having the mouse sirloin.
Some of that, yeah. The hair, the connective tissue and stuff kind of will be measured as animal fiber. And so that's kind of what comes into
like
it.
If you did a total dietary fiber type analysis, you would, even
yeah,
though it's
so
not
you'll measure
carbohydrate, you'd get a reading out of it that there's some fiber in there.
Yeah, so it'll measure as fiber, and even some of the, like in insects, the chitin and things like that will measure as fiber as well, even though it's coming from animals. So there is on the carnivore side, it's almost all just cat research basically. I think they'll talk about animal fiber a little bit more and where it might come from, and just providing a little bit of laxation. Where again, it might not, it all depends on how much is there, of course, what the diet is, of how much fiber it measures as, but you get in certain, um, chicken byproduct meal or something like that, if there's more connective tissue there, a lot of that connective tissue will be not digestible,
Yeah.
and it might not be fermentable either. So it'll measure as fiber in our,
That's
the
amazing.
TDF assay.
It's like almost
So
like it's cellulose.
yeah. So you come down and sometimes when you do all the assays, you're like, oh, we added up to, sometimes it'll add up to more than 100%, and you're like, why? Why does it do this? And usually sometimes it'll be the animal fiber that'll kind of add. So if you
Yeah.
just cook it, or depending on what enzymes you have, it'll measure as protein, but it also will measure as fiber. And in some of those assays too, if the fiber is bound to other things, like you said, in a carnivore diet, you wouldn't measure it, but in other analyses, if the fiber is bound to other things, there might be more measured as fiber than not. If you're just using more of a simplistic assay where you're filtering things, I know we're getting into methodology there, but that's always the trick of, okay, it's supposed to have this much fiber, and then we measure it in the lab and it has this much. Well, why, or where is it coming from, and what is it? So all these methods are a little different too, and that's the difficulty. And there's a term too that kind of relates here too, is you I think it was Justin Sonnenberg. I think he was the one that coined it, microbially accessible carbohydrates, so MAC or max. The problem with that term, or the biggest problem I see, is there's no assay that will allow you to measure that. So it's very messy. That conceptually, it's a great idea that what is non-digestible, what is then fermentable to the bacteria, but you'd have to do many, many assays to, in many different ways, and probably an in vitro fermentation actually, to see what is fermentable, what can we measure as fiber, and then do some math to see what that number would actually be. But it's great conceptually, but what you can measure in a laboratory is very problematic. And anything that you're like, if you're a food or feed supplier or ingredient supplier, you're going to be measuring something in the lab. Yeah. You're not, routinely, you have to have something you can routinely measure in the lab, and then guarantee that on the label. And so, you're not going to do this elaborate 17 assays just to get our fiber and to validate that. You're not going to do that. So, I know I'm getting off onto another spot there, but that's the tricky side of, I guess, with human foods, but also with pet foods. You're guaranteeing a maximum fiber is what you have to guarantee, and it's complete and balanced food. So you want to provide fiber, and that's where some people get on the cat side. You could argue how much fiber do they really need, because they didn't evolve eating much fiber. Even if you can argue they eat the whole mouse, and there's fiber within the gut of that mouse if they're eating vegetable, plant-based products. So there's some fiber there, but still, it's not a huge amount of fiber that they would be consuming, kind of as consuming a whole prey. So there are some people that argue about that as well, of how much carbohydrate and just fiber should be in a cat food.
The fiber that does go in cat food, is it mostly insoluble then, or is it soluble as well?
There will be some soluble, and that's where it's kind of the Fahey rule, and I think over years of experience that he usually says, if it's about 75%, 70 to 80% insoluble, and about maybe 20 to 30% soluble, that's a pretty good ratio. And the main outcome of that, I can tell you, we can do all the elaborate testing we want to, but the main outcome for that ratio working is stool quality, because that's what the most pet owners, that's what it comes down to when they think about fiber and if it's a problem or not. It's usually a stool quality. Of course, it's like any calcium phosphorus ratios or something, the amount, or think like an omega-6, omega-3 fatty acids, it's not only the ratio, it's the amount of would be the amount of fiber that's there as well. So I think in the cat, it probably, there's going to be a threshold of how much soluble fiber can they handle, like how many percentage of their food, or grams per day, or there's got to be some number that I don't think anyone really has a magic number that they have in their head, but that 3-to-1, 4-to-1 ratio does work pretty well, insoluble to soluble fiber. But you just have to keep in mind too how much fiber is there as well.
That brings up that's an excellent comparison to make, which comes back to our discussions on food matrix, ratios of like omega-3 to omega-6. So in a diet, you could also have a ratio of soluble to insoluble. So if you're trying to extract fiber, whether it be soluble or insoluble, feed that alone, that could have a very different effect based on if it was in some other ratio to another fiber, which is a scary thought from the perspective of trial design.
Yeah, you'd that's what you could do. I know most people will do, if you do a dose response, see how much we could do, but how much can we give in the diet? How do they respond? But yeah, you could do a completely, well, you could double your study, or you could do two separate studies if you really wanted to get to, if I want to look at different fractions. And we've looked at fiber blends before. That's what people are going to as well in pet foods, where some fibers, and beet pulp is a good example. Some of these vegetable fruit pummices are decent examples as well. By on their own, they have a nice mixture of both soluble and insoluble fibers. But other people want, either due to patent or due to, they want something new. They want a blend, and so they'll have a fiber blend. And so a key part of that is, how much can you put in there, and what do you want to use as part of that blend? What is, either from a marketability perspective or maybe availability, or can they get a good supply of it? There's many variables there, but so we've done some in vitros looking at a couple on their fibers on their own, and then now let's combine them at a few different ratios to see what those kinetics will give us, and then ultimately then you'd want to move into the animal to see how they perform. But that's when you think about fiber analytically designing studies, what's on the label, there's so many different ways. It gets really complicated, so you have to think about what's your main goal first, and then see how you do that, because it can get really complicated really quickly. So you really have to know what you want to do before you just kind of jump in, because you'll have 20 groups and all these factors that you can't control, and it'll be really difficult.
Got to make your world small in some way.
Yeah, you have to.
That's where I can't remember if I've already talked to you about this, but when I was researching ingredients or just nutritional components of infant formula and breast milk, DHA is fairly well researched. There's, of course, more research to do, but I think of the nutrients that might impact brain development in pediatric nutrition, DHA is up there, like top 5 probably. And there were some reviews done a while ago now. There probably might be more recent ones. I think this one was maybe done 10 years ago, looking at DHA and ARA. And they found that DHA was only beneficial for like eye and cognitive health at a certain dose, but then it also had to be at a certain ratio with arachidonic acid. So that made it really tricky, because you'd look at all these studies on just DHA, and they have really different physiological effects than if you had them together. So, and that's just 2 molecules. There you have 2 molecules and 2 doses, I guess, and you have to have you've got a little window there of success, or to at least demonstrate that it has some physiological benefit. So then there probably is, not probably, there's definitely more complexity in terms of maybe like other lipid structures that those fatty acids might be attached to. But bring that type of comparison back to fiber, and how many different little characteristics have to be just right to get that physiological benefit. And you mentioned feeding blends. I mean, that's probably wise. It's definitely wise from a patent perspective, because someone else it's probably crowded in the sense of being able to create a new fiber. People have done that for a while, or create a new method of making that fiber. People have done that, but you can put them together in potentially novel ways. Which, of course, from a scientific perspective, you're just like, you're making this complicated for us. Why do you got to go with a blend here? There's too many things.
Yeah. Well, you said something. I wrote one thing down, that something, maybe, I'm not sure if it relates it right here, but when people talk about, people forget about fiber, but they talk about, fat, carbohydrate, protein ratios. Usually they're carbohydrate. They're only talking about starch. So on the obesity side, when I do obesity work, and I've asked people at microbiome conferences, a high-fat diet did this. And so I'll start asking, well, if that went up, what went down, and where is fiber at? Because people usually they totally ignore fiber.
Who cares about fiber?
You could have the effect of a high-fat, low-carb diet would be very different depending on how much fiber you have there. So, because when they say high-fat, low-carb, it's usually starch is what they say, carbohydrate. It's always digestible carbohydrate. And so it drives me crazy. But so that's the other thing, like you said, if you just focus on one category and ignore the others, you can say high fiber, low fiber. But of course, that would if you're focused on fiber, because we're focusing on fiber here, the fat content would definitely impact what the effect of a high fiber or low fiber diet would be,
Mm-hmm.
because if it's high fiber, low fat, of course, would be very different than high fiber, high fat. And, so there's only 100%, of course, you
Yeah.
have to get up to 100% somehow, but it really becomes complicated. And sometimes some studies are designed where there's a huge bias,
Oh.
and and again, there's only so many things you can control when you get to diet. What stays the same, what has to shift, but um, that's just it gets really tricky if you're just one dimensional. We're looking at fiber, we're controlling fiber. There's other things can be moving around, and it's completely will kind of blindside you. And it's the worst if you don't see that until you're trying to publish it, and then this reviewer comes and says, "Whoa, whoa, whoa,
Yeah.
wait a second. This is not
Oops.
legitimate comparison because this other factor is there that you haven't thought about." And so it gets to be it's difficult. So you try to think about it in many different ways, but yeah,
food's just impossible. It's just you can't do
yeah,
it.
it's complicated. So
I don't know if you were on any of the committees, but I know like Hannah Holscher has had some students. I think both Hannah and Naman Khan have had students studying avocados, and they've looked at their dietary intake, and it's really difficult to study a whole food because I know they designed menus, but as you've said, things have to add up to 100, so something's got to come or go. And so you could look at what people you could look at in RCT, people who are controlled to eat avocado, and it's still hard to make everything the same fiber, the same lipids, the same protein, the same starch. You could go the other route, and you could look at cohort data, but the type of people to consume avocados just also consume other things. So it's like no matter how you design a controlled trial or do an observational one, it's like, okay, well, are these effects because of an avocado or because the macronutrient profile changed, or because because they started feeling more full and had more or less of a different type of fiber, which is just this like paradox of like you can't be reductionist, but you have to be, but you also have to take this holistic. I don't want to use the word holistic, but maybe well-rounded view. And
no, you're right.
Yeah.
That but it depends on the variable that's the most important. But I was just on a thesis defense exam earlier this week, and so there was the student was looking at, they wanted to keep digestible carbohydrates the same, and they were looking at different kind of different dietary inclusion levels of different ingredients and everything. But they were very focused on that. But as it turns out, the way they were adding things, as you went from, I'll just say from A to E, fiber went up and protein went down. So if you're making claims about certain things, you're saying in these ingredients, I said, but okay, but diet E had a lot more fiber and a lot less protein than what this other one had. So you can't say alone it was due to that ingredient. It was because the macronutrient profile is very different, and probably micronutrient as well. So when it comes down to research, is what variables are you going to control and what other variables maybe you can't control, but hopefully if you can acknowledge the other things that happen. And while you're designing the diets or your outcomes, you really have to think about what other things might be changing. Hopefully some level, because you can kind of invalidate at least what you're trying to prove if it's too different in another way. Some ways it's easy, like you said, if they were just on that study. I wasn't part of the study. I've been on a couple committees, I think, with students that were kind of involved. It was a really big, elaborate study. If you're to say adding an avocado or adding anything, an extra something a day, and just go with it that way, just say you're just adding it. But then you could say, what if you add it and then you try to make adjustments? So again, you're adjusting for fiber or fat or whatever. You can do it that way too. So you can make an argument for both, I think, but depending on how you design it really affects what you can interpret and what you can say about it at the end. So you really have to know what you want to say at the end. If you're on the marketing side, what's your claim going to be, or what's your angle here? Because if you design it the wrong way, then you clearly can't say it. So you can't give that claim because there's other factors changing. So
since we do a lot of literature review nowadays, I kind of feel less worried about like the good design of an individual study and about groups of them, because sometimes a clinical trial is designed really poorly for one question, but actually well for another question. But you got to make a
Yeah.
story around your data and publish it somewhere, so you're going to go with some sort of story. So on that note of like what you're going to replace, so the European Food and Safety Administration came out with this draft opinion on the the tolerable upper intake of dietary sugar. And you can think of sugar as being total, free, or added. Total just being everything that's in that food. Added typically meaning it was added during processing. We're not even going to talk about free. And they had found that there was a relationship between how much added sugar people consumed and adverse health endpoints. Think of like diabetes, gout, metabolic syndrome. So, okay, more added sugar equals bad health outcomes. But then they have what I thought was like should have been bolded at the very top of everything is if you look at added sugar isocalorically exchanged for other macronutrients, that relationship went away. So if you look at it on an isocaloric comparison, some amount of added sugar versus some amount of any other energy source, could be starch, protein, fat, it no longer had this relationship to disease. And I think there's this, there's a lot of discussion right now about food quality. Is calories in, calories out versus this carbohydrate insulin model. And I think it's a pretty big report. I think EFSA did a fairly good job. And I thought, wow, this just seems straight up calories in, calories out here. And I think that's a hill for some people to die on. I'm not going to die on that hill. But that was extremely important then to you. And they have like all the forest plots with their meta-analyses. You can look at, not only can you look at a food in isocaloric exchange with another, or isolipidic, isonitrogenous, whatever. What about the diet overall? Was that meeting the energy needs, or was it an energy surplus or energy deficit? So the two food thing comparisons have to be, well, they don't have to be, but you should at least know if they have similar energy or whatever. But then you should also look at the overall diet. So to your point, like, okay, you could design that study to just add an avocado, or you could replace it. It probably has to be done both ways.
The outcomes, I think they do differ a little bit depending on how you design it. What you can say. No, that I don't know the specifics of what you just like on the added
Sure.
sugar,
No, that's just an example.
Point. But yeah, because it does matter that how you represent the data.
It's complex, but it's not at the same time. It's like, did you have the right covariates in place? Okay, well, if you didn't, then we gotta think about this in context of what you did. No, it definitely, the design dictates what you can say or can't say about it at the end. So it, and you, again, every study you can't do everything in one study. So you have to really think about what are your main outcomes and why, what do you want to prove or disprove, and then, design it the correct way and then do it. But it's a lot easier. Does this sound so like haughty-taughty and arrogant? Oh, fiber's so complex. It's a lot easier, I think, to design a study where two groups have similar fat, protein, or carb. You could design them with similar fiber, but as you've talked about this whole time, like, do they have the same ratio of soluble to insoluble, or what type of fiber was it? I suppose you can make the same argument. What type of fats were they? What type of lipids? What type of proteins and things like that? So, I'm also tired of seeing those studies where it's like, we fed animals a high-fat, high-carb cafeteria diet. And you could actually look at what's in there, and I'm like, is this relevant? I don't know if anyone gets that much of their energy from straight-up sucrose, but okay.
No, this is where the arguments or discussions we can decide what each one is probably separate. But even when I was a grad student, I was at a conference and people were presenting this big, I won't say names or anything, but it was published at this paper was published in a pretty big journal, and they were from a very prominent group, is all I'll say. But all their data hinged on crude fiber, which is a horrible measure of fiber. It's a very poor measure of fiber. So all of their correlations and all this assessment was done that way. So within our group, the paper had come out before the conference, and so I was, I was the grad student
Ready to storm up to that poster and just
a few hundred. Well, I was in a plenary talk, like hundreds of people in this room, and I got the nerve up to go to the microphone and ask the question and kind of discredit the expert in the room, but it was a complete. In fact, he still said when I got back, you were right, and you
did it backfire. Well, how did that play out? Did it backfire on you, or did they go, you know what, you're right?
No, they kind of. They weren't fiber experts, and that's part of the problem. That they didn't know what they were talking about on the fiber side, but they're making all these claims about fiber and health, and it was like, oh, fiber shows no benefit for X, Y, and Z. Well, yeah, if you don't measure fiber accurately, you're not going to show a benefit because it's not being measured correctly, like anything. And so, even today, though, and so that was, that's 20 years ago. And so they kind of say, oh, we used some measure, but they didn't really know what was used actually, as it turns out, which is a poor measure. So anyway, I've thought about that many times, like, oh, I got the nerve up to say what I wanted to say, but it was always, once I sat down, I'm like, oh, I should have said one more thing, it's one of those things. But still, people still think of, unfortunately, like you said, people understand, oh, there's different types of lipids, there's different types of whatever. When it comes to fibers, there are a lot of people that still say just fiber. They don't know all the complexities that we've been talking about for the last 90 minutes or so. Yeah. On just saying fiber does not mean fiber. They have all these different properties, and it still gets to me sometimes that, oh, the high fiber diet didn't change the microbiome. Well, if depending on what it is and what you call high fiber, number one, what is high fiber and what changed? But then also, if it's pure just cellulose,
that's high fiber.
It might not change it at all because it's not being fermented, so it depends on the whole situation. And so sometimes people will say, oh, fiber doesn't have an effect, but it depends on the type of fiber you're talking about.
Wild thing to say. I saw articles like that floating around on LinkedIn too, of like, I don't know what it was, but it came out recently, and it was like, fiber doesn't affect the gut a lot, but prebiotics do. Or maybe it was about fermentable foods, and I should go read it before I make any sort of comments about it. But it's really, I guess I understand it in some ways. You gotta your title's gotta say something on that paper, but just to say fiber is a little too broad. Food does or does not change.
Yeah.
But and to your point earlier, like, what were those subjects consuming beforehand? What was their microbiome used to, and that could have a big impact. So yeah, I get like aggravated sometimes by these headlines too, and I'm like, I shouldn't take it too personally.
Yeah, no, and that's part of it. Is sometimes a lot of in the media you'll see the headline has to sell, right? So that But some of the publications,
you're right.
Some of them want a definitive statement, where others, no, you can't say anything about a benefit or a detriment. You just say, tell me what you studied, and they have to read it. But some of them are very kind of forthcoming that, fiber does nothing, or fiber is the best, but
pick a side.
It cures everything. Like, well, it doesn't. But
even my own research, we looked at safety of pectin and formula and feeding it to pigs, and we didn't say safety of high molecular weight, this type of pectin from this. Your title becomes ridiculous, right? We at least put a dosage in the title, but I could have gone, if in retrospect, had I known. Well, I suppose this is what the method's for. We at least said what it was. We've read studies where they said we fed pectin, and I'm like, I would like to know what kind, please. Can you give a brand so I could look it up or call someone? Not just straight up pectin. We fed cellulose.
Yeah. No, that's where too. I spoke at an NIH conference a few years ago. I was on the microbiome, and it was kind of methodologies and stuff, and my task really, I was supposed to talk about dogs and cats, but or non-rodent models, I guess. So I spoke about dogs and pigs is what I spoke about. But as part of, there was another, it said, speakers, if you can answer some of these questions. So, but part of it was about diet, and I kind of got on my soapbox for a little while, and I guess I ended up preaching to the choir, but there are a lot of people that thank me afterwards that I said, we are so specific about all these methods, about what we measure and what we do. But then when it comes to diet, we'll just feed that diet. And so like, no, you can't do that. You have to think about what fiber, all the components and, a lot of the stuff we've been talking about. But a lot of people thank me afterwards because you read all these papers that they don't give a level of detail that you ever could replicate anything. And when it comes to diet, like you said, pectin, it's a big difference, the low molecular weight, high molecular weight, and their properties and everything, how they'll respond. And it's the same, all the way through really for diet composition. Is it a semi purified diet? Is it a what kind of ingredients were you using? Because all these things, particle size and everything, the matrix is completely different. So if you haven't described any of that, well, then it really doesn't advance the science because you can't replicate it anyway. And you don't know which, if you're going to buy a special ingredient, you have to know what it was, unless it's labeled, like you said, what's the source, what's the catalog number or whatever.
Even then though, there's no, it's not for sure that the manufacturer is going to have all that information.
Yeah,
or they might list like,
hopefully...
it was 40% pectin, and you're like, but what else is in there? And I don't know.
Yeah, a lot of those ingredients will guarantee something,
right.
But the
What's
other
the rest of the stuff in there?
You don't know what's guaranteed. What is that? Because it's not well defined. So
this is the difficulty though, because we come across this issue just in publishing where probably everyone who's best suited to review my work, I have some sort of conflict of interest with, whether negative or positive. It's just like, well, I've talked to them here and we've worked over there. So I end up getting these reviewers who don't know anything about like the intersection of all the different fields we work in. Sometimes honestly I'm like, well, those were easy reviews. And then other times I'm just like banging my head against the wall. I'm like, why are you even asking these questions? These are ridiculous. But like, do you get that in your own work too? Where it's just like, well, I'd love for this person to review it because they are intimately aware with what would be good and bad. But like, I've published with them before, or we're on the same committees. Like, there's just too much bias.
Yeah, that it is, especially in the microbiome space in particular. There's a lot of people, but even, I tend to be fairly collaborative, so I collaborate with a lot of people. So then you get to a point where, I guess I don't put them on my recommended reviewer list because I don't, even if I think they could do an unbiased review, or else if it's been a few years, then I think, well, I haven't worked with them in a while. Maybe it's okay. But I've heard other people say that too, that if you publish with a lot of people, who ends up reviewing your papers, and sometimes they'll, like you said, sometimes you'll get almost the extremes. You get one that then they completely don't understand what was going on, and so, or what was designed, what the intent was. And if it's a dog and a cat, sometimes they'll say, this has nothing to do with human nutrition. And I say, exactly, it's not for human nutrition, it's for dog and cat nutrition. And so that you get that rejection because they're thinking, oh, it's a model, but you haven't. You're like,
no, it's a model for itself.
It's a model it's a target species. It's not a model.
You're right, yeah.
And so that's very frustrating. But then you get the others that they clearly, it's an easy way to publish, I suppose, because they didn't, like, it just goes through almost without any comment at all, which I think, well, that's not true either. Like, it wasn't perfect. So you can add something, and I guess those reviews are easier to deal with because then you're published. But
I love those where it's no comment, and they're just like, quality of writing, 2 out of 5. Novelty of this, 3 out of 5. And you're just like, but you've got no feedback for me.
Yeah. No,
just lukewarm all the way.
Yeah, no, that's, whether it's good or bad, there should be some constructive feedback of some kind. Yeah, that is a tricky thing. And I think now too that people are just overwhelmed with how many things are going on, whether it's COVID or not. So a lot of the good reviewers won't review because they're busy. They're asked to,
you could spend your entire job just reviewing papers all day.
Oh yeah. So, you could definitely, in the fiber microbiome, that space, you could just, that could be a full-time job. So it's tricky, but that's the frustrating part, that then sometimes the reviews, whether it's positive or negative, you don't have much to help you. A really good reviewer actually will, even if it's a positive review, they'll still add, say, I would still do this or clarify that, because you might think you've described it well, but you haven't. Yeah. So you should, clarify this. And so then, a good reviewer will improve the quality of the paper, because you might think, oh, everything's great and this is the best draft we have, but you're missing something, so
you hit submit and you're like, this is perfection. And you get reviews and you're just like, wow, there's just like typos and words missing. and like
Yeah,
10 people
yeah,
reviewed this. We all got our blinders
yeah,
on.
yeah, oh yeah, yeah. So yeah, things, and sometimes if you, especially if you're the main author, you know what you want to say, so you just, you don't see some of that it's just not there, but yeah, but it gets to be tricky sometimes of getting in. That's probably goes for every field, I guess. Is just getting an accurate reviewer that's an unbiased source and getting it through. But yeah, sometimes you'll see, especially you'll see the higher journals. Sometimes there'll be huge flaws when it comes to diet. That is like, if you went to a nutrition journal, this would never got published. But now you're in Science, and you got the reviewer that, based on your name, largely, and the reviewer that didn't know what they're really
Mm.
talking about got it accepted, and that can be a problem as well. That repeating, oh, so and so, they'll cite the paper, but repeating it doesn't happen because then you get people in the nutrition side that know what they're talking about.
Yeah.
It doesn't work, and so they can't replicate it. So again, that's just the frustrations with publications, I guess, in general. But
I guess it's a really bad format to use to talk about studies and data, because you can only say so much in a limited place, and
yeah,
there's too many. Like if you do a trial on humans, you could say, they were on average 30 years old. Give a standard deviation. There's so much more complexity to that. The distribution and was there a relationship between how old and the sex they were, or ethnicity, or even just basic things like that. But you've only got a table to describe those things in, and I guess I don't have a better solution. I mean, we are at a time now where you could almost make interactive documents online, but I don't see that happening soon, or like ever.
It's tough. Yeah, the biggest thing is providing it as much description in the methods as possible. Yeah. I guess from like our grad students, we try to get to that level of, if it's a special ingredient of some kind, what's the source, and what, can we describe it beyond just saying it's this,
beyond go see supplemental.
Yeah.
Described previously.
Yeah, exactly.
Where is it?
In a citation that may or may not be relevant from 20 years ago. Yeah, you're like, you need details here. That's where I guess from day one, I've always been just kind of beat into my head that the diet matrix, the diet composition is very important. So, and not only what you predicted, but analyzing your diet to confirm that what you thought you had is what you have now. And so, on the animal side, on the humans, it's, you do the best that you can with the dietary records, but it's, you try to confirm, educate them up front, and then trying to check in with them periodically. So hopefully you say, back to my course, and they do a 3-day dietary record. I'm like, did you really eat this much, hummus? Or, because they're trying to say, I ate this hummus and here's how much fiber. And it looks like, I think that's the entire package. Did you eat the entire container of this?
Joke's on you. They did. Yeah.
But to get your 30 grams of fiber at one bolus or not, whatever the number would be. But there were a couple of people that kind of were overachievers. I'm like, I don't know if I believe this dietary record, because what the numbers you have here are very hard to believe. And so, you'll see that on the animal side though too, that if you're not in some studies, they won't measure intake, they won't measure like fecal output. So then you make some claims, and that was early microbiome studies that, especially when you think about fermentation and obesity, saying, oh, it was highly fermented, they produced short chain fatty acids, and now they're obese. But what was food intake? What was, if you look at the energetics behind it, a lot of that was never there. And I'd speak to the authors. I'd seek them out at conferences and say, not, it's a personal conversation. I didn't go online and say anything negative, but I said, did you measure that or didn't you? And I almost kind of say, if you don't measure that, if food intake is the main difference, well, then you're attributing it to X, Y, and Z, but maybe they just ate more, and that's why, or they ate less. But it's very simple, or it's very easy to get very, very molecular and very complicated, when in fact, sometimes the answer is very easy. So usually for studies, I'll usually start at the easy part. Oh, sure, we can measure the microbiome, we can do this, but we better have some relatable measurables that make sense, like just health-wise, before we just don't bypass those to go to the very, because people want to see microbiome data and metabolomics data, which is great, but you better have the other ones along with it, at least, so you can relate it to their overall response.
Absolutely.
like stool quality and stuff like that. And sometimes it's easiest to do that first. Do a pilot study or a smaller study up front to see where you're at,
Mm-hmm.
to get your dose in the right range, and then deep, dive in. Sometimes you can design it and do all of that, but it if you really don't know what you have to deal with at all, I wouldn't
If it
you wouldn't sink that much money into it, unless you really are confident you think you know what you have there. So
Yeah, if it's, I mean, maybe aside from really wanting to understand basic or foundational research, if it's not going to move the needle on whatever endpoint of interest you have, then, I mean, do you want to look into the mechanisms to how you got to nowhere?
yeah, that's what. Yeah, you want to know is there an effect first before you start looking at a mechanism? Because
Yeah.
if yeah, because you'll sink all the money into measuring things, but then in the end, yeah, it didn't change anyway. So they're a mechanism of what?
Well,
Because
yeah, yeah.
the outcome didn't change.
Well,
So
and you can look at it like you can. I guess that's what perspective you're looking at. You're looking at it from the outcome or from the input. Because you could go, okay, well,
yeah,
fiber, it's soluble. Okay, maybe it has gelling properties. It's also fermentable. And I know that when things are fermented, short chains are made. And I know when those are made, this happens. And so you'd just be, I
mm,
feel like you'd just be stuck in the intestines forever for your entire life before you even got to like, how did it affect the rest of the host? I suppose you could do the
yeah,
opposite and you'd be like, I don't know, we fed them fiber. They did well. They did pretty good. I don't know how,
yeah.
but they did good.
Yeah. Well, that's what oftentimes it is. I'll do the top down initially or at least say, do you, from the top down view, this much fiber, does it show, is there a difference in this or that? Simple things, food intake, stool quality,
Mm.
in fecal short chains, you could do something like that where it's not, you can argue about the value of fecal short chain fatty
But that's
acids
low
because
hanging fruit, right? I mean, it's not
but
hard to do.
still, if it's way more fermentable, you'll see changes in short chains in the feces, but that's a. And then I know in some of our correspondents too are thinking about other outcomes, not just laxation, blood
Mm.
lipids, blood glucose, but you get into more on maybe the mood side, the gut-brain axis. Those are very complicated, but you'd want to see if you can show some measurable
Yeah.
thing at whatever you're measuring an anxiety to be or anything with cognition. You have that effect before you start looking at the mechanism,
Man,
to
I
make
would.
sure you
Yeah.
have a pretty good foundation.
I think you.
But then even there, the gut. I mean, I know you have nerve. The vagus
Yeah.
nerve can do certain things. There can be, short chain fatty acids in the gut peptides. There can be other, it gets very complicated to prove those mechanisms, and that's where I struggle. And like you said, will there be, FDA? Will there be some of those measurables as being maybe a health benefit in the long, in the long
Mm.
term? They might be, but it takes a lot of,
Yeah.
I think, a lot of data to lay that foundation to make, and then the right studies designed in the right way to make it believable. Because laxation by far is still laxation stool volume is still the easiest thing to say to show. Yeah, this is a fiber.
Yeah. Yeah.
To go beyond that, it gets. I mean, you can do blood lipids, blood glucose. You can show those as well. You get into satiety,
That's
then it
tricky.
gets what's your measurable. Even laxation, there are people that will argue. Do you look at stool, bowel movements per week? Are you looking at overall stool volume? What's your measurable?
Yeah.
And so you have to make sure you know what you want to do, and then design the study to show that allows you to show that. If you're not, but those can be quite invasive though, to take complete, everyone's stool output for a week or something like that to get an actual reliable
Yeah.
value that, just given the variation in people's food intake, I'll say their bowel movement frequency,
Yeah.
it differs. And so it's just, you can't just say, oh, we just want one day, and
Oh man. Yeah.
you know it doesn't work. That you have to have the study designed in such a way to do that. But it becomes more intensive with the subjects and retaining the subjects over X amount of weeks or months or whatever you have. And so
That.
it
Yeah. That.
yeah, it becomes difficult there to show that benefit. And on the cognition side, I'd argue it'd probably more complicated there to what's your measure, what are your outcomes, because it's quite variable and subjective
Yeah. Very.
with a lot of them. From what I understand, I'm not in that area, but you listen to the experts, and some of them, if they're doing a meta-analysis on something, those measurables are very difficult to kind of just throw together and say,
Yeah.
oh, because all the studies are designed and done a little differently. And can you group them together and say, collectively these say this same thing or
Yeah.
not,
Yeah.
or can't you? So it's complicated when you get to, um, and there's just so many
It's
things that have to happen from the gut to the brain,
yeah.
or can happen.
Yeah,
It's difficult.
there's a lot of places that you could lose an effect or that it could have one. And I mean,
Yeah,
cognition, and because I was on the neuroscience side of the lab in grad school, I mean, it's really easy to be like, oh, it's so complicated. But I got tripped up by simple things like short chains because I would read, just the things everyone would say about them and what they do, and then I'd go, it feels like there's a little more complexity here than I realize, and there definitely is. And sometimes, maybe short chain is a bad example. No, it's not a bad example. We looked at like the variability around that. You could look at coefficient of variation and things, and there were some studies where like this is unexpectedly high, and we have no reason to believe the method was done poorly, unless it was, and we just didn't notice or something, or didn't report it. And on the
Mhm.
cognition side, it's very easy for people to be like, oh, a lot of things are self-report, which is subjective. That's true. And there's a lot of variability. I'm like, there's a lot of variability in just innate biology too. Like you're trying to make it sound like all this brain stuff is specific. It's not. Like blood glucose,
Yeah,
that itself, there's a lot of variability in that.
yeah,
Um,
yeah.
and that what's probably unhelpful for a lot of manufacturers is that the FDA doesn't have, I think probably rightly so, they don't have great definitions on what a health benefit is. Um,
Mhm.
because
Yeah,
as you said, like proving laxation itself can be difficult. So try to prove that someone feels better or has less anxiety or depression. Like there are measurements of those, but they can be tricky, and you might open the door to too many people trying to make crazy claims. Um,
yeah,
but we,
yeah,
on
yeah.
the laxation side, we helped a client with a clinical trial where they were fed a product high in protein and fiber and looked at. We've actually got three measures of laxation. We've got a bowel habit diary that they took for 3 days, 6 days leading up to every visit. We've got oh my gosh, the GSRS, gastrointestinal symptom rating scale. And
Hmm.
then we also have, I guess part of the bowel habits diary is we do have Bristol stool scales. So we're trying to
Hmm.
understand, these people wrote down how many times they had to strain at the beginning and at the end. Did they have to have
Hmm.
a laxative? What was the Bristol stool scale of them? We've got
Yeah,
gastrointestinal reports of like pain and discomfort and constipation. And so we're just trying to understand, like, do these two things even align with each other? Do people know when they are constipated or when they have diarrhea? Um, and then the it ended up going into the supplemental. What ended up happening is there was no difference between groups in like frequencies or rates of bowel movements. And some would probably argue we're about to go on a fishing expedition, but now we're trying to understand, like, well, is this only helpful for people who already have diarrhea or who already have
hmm,
constipation? So it's like drilling down into that, to those subject populations more and more, and say like, okay, if you're already healthy, this may not matter. It's really for those people
yeah,
who have that baseline level of constipation or
hmm,
loose stools. So, just
yeah,
slice and dice your clinical trial until you have like zero statistical power anymore.
yeah, yeah, yeah.
We started with like
Well,
100
that's
people,
where,
there's like 10.
yeah, yeah, no, that's where I think, definitely you have to recommend whether it's on the pet side or human side. If you're really in the end want to make a claim, you really should talk to FDA and say, because it is their answer oftentimes, well, it depends. If they're on the, especially on a podium at a conference, it depends, and it does depend,
Yeah.
because what are you trying to show, and what are your measures, and then can you then say A, B, or C, on that. But it depends on a lot of factors. So oftentimes you'll share, here's our study design, this is what we want to do, these are our outcomes, this is the population. Because to your point too, that I've heard questions before, and can you have a population that's slightly constipated, in quotes, that, so they're
Yeah,
not a
yeah.
clinical case, but it's, again, it's going to be easier to show a change in bowel movements if you have a population that's not clinically, ill,
Mhm.
but that they have a lower frequency than those that are very regular, of course you can't. How are you going to make them more regular?
Yeah.
So it's hard to do. So you really have to think about a lot of factors, and then I would really encourage. I always talk to FDA, say, here's our plan. I know it adds, might add a few months on the front end, but then you, and usually they'll. I know on the pet side, certainly they'll say, here's our for a new ingredient, or you want to do something there from an efficacy and safety side. They'll look at it and say, well, I would extend it to this long. You only have X amount of weeks. You really need to push it out this way, and then they'll kind of sign off on. You're still never guaranteed if the response isn't what you think it is, but if you just say, you know what, I'm just going to do this study and this is what we're going to do, but then you don't. FDA only sees the results afterwards. They might have a huge problem with your design that would, and then you've wasted all that time and energy. So, yeah, it's but like you said, sometimes you can take a subset, and we've even done that we did on a agave inulin where we looked at bifidobacterium population changes, just relative abundance. But what it ended up being was almost every woman on the study had a nice response, and men were kind of hit and miss. So either it was due to dosage based on their kilogram body weight or their amount of food that they ate or whatever it was, or just something inherently hormonal different between the men and the women on our study. But things like that too, that and nowadays we've done studies in the past. We were worried about I'll just say that the estrus cycle and how that might influence certain things. We did a study where it was just men. Well, then a few years later, we went to IRB, and so I was like, why are you only using just men? And
That's all over on animal stuff. They're like, we don't want to deal with estrus cycle. There's too many variables.
Yeah, exactly. And so then you're like, well, why? So in this study, and even the sponsor, they wanted, we need to apply this to all people, not just men or not just women. So we did it that way. But then, but there's that variance, and whether it's just due to like body size and food intake is one thing, or depending on life stage too, if it's pre or postmenopausal, just that alone, there's estrogen's effects, and kind of the estrogen can affect the host and the microbiota, but also the recycling of estrogens can be impacted by the microbiota. So it's like this, give and take a little bit, that they can affect each other. So it really, you have to think about all those variables as well to make it even more complicated. But it's
too many things to consider in designing a clinical trial. You're like, it's fine. We'll have like two groups. Maybe they cross over. Don't get your power analysis done. Hit the ground running. It's definitely more work this way. But I think maybe instead of just saying it's too complicated, maybe some of the simpler solutions I think could be to also measure some of the health outcomes during the inclusion or screening process, which I don't know, maybe that's a double-edged sword, because then you just go like, oh man, none of these people fit the inclusion criteria, so now we don't have a study to do. But it would probably be helpful though, make sure you've at least targeted the right population. But especially nowadays and in pandemic times, it's harder to recruit and I guess basically do anything.
Well, we did it. We with Hannah Holster and one of her postdocs, one of her grad students, we wrote a meta-analysis, a systematic review, not a meta-analysis, a systematic review on inulin type fructans, and it was kind of for many different outcomes, and definitely BMI and age were key parts of that. That anyone in above, I think it was above 70 or 75. Now I think it's 70, which most of the studies were in that category anyway. But we're looking at bone mineral density and things like that. But the focus was really adults. So a lot of that data is in adolescent girls in particular. There's some data still in adults too. But the criteria, if you're doing that, really, who are you talking to, and whether it's a systematic review, a meta-analysis, or you're just talking to FDA regulators on what your claim is going to be, it's, well, who's this intended for? It really, you have to think about those guidelines a little bit as well. And so it all kind of comes into play. So you can at least say something to the population. Usually you're targeting adults, but it depends on the product. You could, target adolescents or geriatrics on each end of the spectrum too, especially on the gut health side. You can easily make that argument sometimes, but then the literature you're using or the studies you're doing has to be relevant to that. So it really there's so many things you can control, but you at least have to think about those. And if you can measure things, measure it. It could be helpful later on in a
Yeah.
post hoc analysis or something, where you're making some correlations maybe, or something that can help explain the data you have. But again, there's this fine balance of, you try to measure too many things, it might just complicate things, or it might affect the other. Your primary outcomes might be affected negatively if people or animals are stressed out all the time. Then you're not,
they're always in my face, just collecting
yeah.
data. Yeah.
So you might not have, might not be end up testing what you think you're testing in, some of those
Your whole
cases,
population is just stressed.
yeah, they're highly stressed because you never left them alone.
Yeah.
So
Well, okay, it sounds like the easiest thing people could do, maybe, without more work, is just to do better reporting. What type of fiber was it? There's the low-hanging fruit.
yeah.
Yeah.
Definitely trying to describe what that was, and if it's natural, then you again, how many avocados was it per day, or how many grams, or something, some kind of measurable. But and then if you can measure in the lab that I'm not sure Hannah and Naman measured that of here's so many avocados, and there's that's in the databases, I suppose. So maybe they didn't measure the nutrient makeup of
I
that.
think they did actually, but
I
see what you mean. I see what you mean. They used
and,
established databases, which is different from also measuring the ones that you have. Okay.
Yeah. On the pet food side, usually, or like animal side, if you're making a complete diet, we always analyze. Is again back to that thesis that defense I had earlier in the week. It was, here's what you thought you were going to do, and then
here's what you actually
sometimes
were.
The ingredients are different, and it ends up being slightly different. So sometimes it doesn't rule out what you've done. It's just, but if you targeted, I wanted 12 grams of fiber intake every day, well, ended up being 14. Well, that changes it. You just have to know what you're adding. So so we always analyze our diets as well, just to make sure we
doubly sure
we're given what we thought we were going to give. So
have that table formulated, analyzed. Please, no one, don't look too closely at how off we were from our formulation. There's no explanation. I don't know. It just is different.
Yeah. Well, and sometimes you just have to own up and say, we targeted this. It ended up being
half of that. So,
and then in your discussion, you can, hopefully it doesn't totally, again, it should be close, but then when you do, when the reader interprets it, it should be, if you formulate it to be 10 and it ends up being 15, in the abstract, you should clarify that, because some people only read the abstract and they'll say, oh, 10 did this. Well,
okay.
And we ended up having 15, whatever the number is, so
can we get our abstract extended to 1,000 words, please? You know what? Just make the whole thing the abstract. That would be the easy.
Yeah. No, it's those can be challenging too. Some journals are more difficult than others to really squeeze in there. It's because, especially if you have complicated designs and a lot of outcomes and a lot of differences, it's really, really, what's most important to have in there, and you're only so many details can go in, so you have to be careful with what you say. But I don't know. That gives you, I guess, the work. There's always a job security there from just getting. I know,
there will
as an advisor,
always be more studies that could be done.
Yeah. How do we get this in here? And you end up doing it, but it's difficult sometimes. But if nothing changes, your differences weren't different. Your differences, or they were trying to. Your hope that it was going to be different. Nothing really changes there. Then it becomes really easy. But then that's for a bad reason, because our intervention didn't do what we thought it was going to do. So back to the drawing board. But
well, we're at our 2 hours. This has been great. What would be the one thing that you think, I guess for someone who doesn't know much about fiber and is working in this space, let's say for researchers on it, what should be like the one thing that the place they should start, or what they should keep in mind?
Oh gosh, I guess I'm a nutritionist, so I'd say, if you can increase your fiber with whether they're enriched foods or natural foods, I think certainly is good. I do think whether it's on the microbiome, whether it's on fiber. If you adhere to the dietary guidelines, a lot of those recommendations are to give you a breadth of food, different types of foods, and getting your whole grains, getting your fruits and vegetables will, if you were to meet your dietary guidelines, that will get you a good portion of your fiber you should have anyway. And I would say, I tell my kids all the time, when you're thinking about vegetables, a variety of vegetables, not just, and don't, I mean, you can get some resistant starch from potatoes, but you're thinking your green leafies, your reds and oranges, and then on the fruit side, have a variety of those. So you're getting different, I think from a nutrition perspective, but also fiber, you can get a lot of that fiber there. But also, if you're buying foods, like we said before, there are some, whether it's pancake mixes or cereals or bars, look at the side of that package, and you can, oftentimes in my experience with some of these soluble fibers, it won't change the taste, or the some of them will, but some of them won't. You don't have to oftentimes change everything. You can make small changes, and it can add up to another 5-6 grams of fiber a day with just a very couple small changes. So you just have to be more intentional, I think, of what you purchase, what you think about what you're eating, I think, and you can do it. So if you want to go hardcore and go get that 28 grams, 38 grams if you're a man, you can do it. But again, you just have to, it's kind of baby steps. If you're eating very low levels of fiber, you wouldn't want to do that all in one day, or you might have a negative response. So you have to build up to it. But it's really, like you said before, whether it's dieting to lose weight or whatever, it's a long-term thing. It has to happen all the time. You just can't have, one day a week, I'm gonna eat a whole bunch of fiber and then I'm not gonna eat any of the rest of the, it's kind of a bigger change from a diet perspective. And there are supplements certainly that can help you get there, but I always think it's best to get it from the natural forms. Even these isolated food, if they're in a food source, it's still better than popping the pill, I think.
Sure.
But I don't want to say anything about Metamucil either. It's very effective, so you can do it that way to provide laxation. It's just ideally you'd be getting them from kind of the natural food. So
have your supplements be supplemental.
Yeah, I think you'd still have to have a good foundation. That's like any nutritional principle. I think that supplements can be useful, but if you have a better, strong foundation, that's always the better way to go about it. So
we got a lot in there. We got like variety is the spice of life. Get everything from like Whole Foods. Look at your labels. And then like baby steps, one thing at a time. Perfect. Yeah. Hey, well, it was great.
It's easy, right? Just take, I don't know, 40, 60, 80 hours to research all this, and then you can figure it all out and then go for it. But no, some of the things are quite simple though. It's kind of simple things. Simpler is better. I think, less is more with some of these that, and that's where you get the whole foods, where you say avocado or, fruits and vegetables. Again, you want to know what the serving size is and everything, but if it's not all, you don't have to get to know everything on the label, what everything means necessarily. You can get to that, a couple key numbers and maybe some ingredients there, and search, find what you're looking for. So, but yeah, it's one day at a time. Just start. If someone, you have to start trying, and then you can educate yourself, and just, if it's online, I also say that too, to make sure it's a reputable source, not just anyone, spouting off. So
anyone can spin up a blog.
Yeah, exactly.
Myself included. Ooh. All right. Well, I gotta hop off from this. Is great to have you on, and I'm sure we'll have many more conversations to come. Hey, thank you so much for tuning in to the Traverse Science podcast. If you'd like more of this content, check out our website on traversescience.com. You can follow us on LinkedIn, subscribe to us on YouTube, and you can check out the podcast audio version on Spotify, Apple Podcasts, and Google Podcasts. All right, take care.